It's a great pleasure and honour for me to introduce the third plenary speaker in this Uppsala EED meeting, Professor Beverley Glover from Cambridge University. Well, Beverley started her scientific career with a degree in St Andrews up in Scotland, and then moved down to the John Innes Institute, where she did a PhD thesis with Cathy Martin, and then went to Cambridge as a postdoc and was recruited to a permanent post, and graduated to professor, as you do, and is now also a director of Cambridge Botanic Garden. So we're particularly lucky to have Beverley here this week, because last week Beverley's passport went missing, which caused a bit of a panic. And it did turn up, but I've been sworn to secrecy, so I'm not allowed to say where it was found. Right, I'd just like to add that Beverley is first of all, Beverley's subject, let's not forget that. So Beverley is a specialist in the mechanisms by which plants attract insect pollinators, and particularly, I think, the ways in which they construct colour through microstructures on the surface of petals and so on, rather than necessarily by employing pigments, as many other crude plant species do. So Beverley's also written an excellent textbook, which is called Introduction to Flowers and Flowering, and I would recommend that you all keep a copy of that on your bedside table next to your passport, possibly.
Thank you, Charlie. So a few of us had a bet on whether he'd be able to do the introduction without mentioning the passport thing, and I'm pleased to say I've just won quite a lot of wine later tonight. So I'm very pleased to be here too, passport notwithstanding, and to have a chance to tell you about some of the work we've been doing in my lab in the last few years. And I want to start off by just introducing the big picture question that we're interested in. I'm going to focus on some of these great flowers and tell you about tricks they use to influence the light in a way that makes them more attractive to animal pollinators. But before I do that, I want to tell you about why we're interested. And so for those of you particularly perhaps who aren't plant people, this is a phylogenetic tree of the land plants. And the thing I really want to pick out on it, so these are our divisions, which are kind of the equivalent of the phyla in the animal kingdom. And I've put species numbers down the right-hand side here. And you can see that the angiosperms, the flowering plants up the top there, have 350,000 or so species. We're not entirely sure yet. And nothing else has anything more than 10, 12,000 species. And plants that you're very familiar with, see plenty of in Sweden, like the conifers, up here only have 700 species. So the plant kingdom is generally composed of groups that are fairly species poor, apart from the flowering plants. And this asymmetry, this enormous species radiation of the angiosperms, is the question I'm interested in. It's a question that evolutionary biologists have been interested in. Darwin described it as an abominable mystery. There are lots of answers to the question, of course, and they involve thinking about speciation rates and extinction rates. And there are lots of very broad patterns you can look at. And one of the broad patterns that comes out when you think about angiosperm speciation and radiation is that their initial speciation rates when they turned up in the fossil record are about the same as all the rest of these plant groups. But the other plant groups' speciation rates drop off with time quite quickly, and the angiosperms don't. They continue to speciate at a great rate. And the reason for that, I think, is to do, or at least in part, with the way they've harnessed animals to move their pollen around, and that's given them the opportunity to engage in reproductive isolation in a way that other plant groups can't. So let me show you what I mean. If you think about a conifer forest like this one, there's an awful lot of species, an awful lot of individuals there in that forest, but there's very little chance whatsoever of any ecological speciation occurring, because there's no mechanism to reproductively isolate, say, this group over here from this group over here. There's no way of preventing them crossbreeding. And the reason for that is to do with the way plants, for the most part, reproduce. On a windy day, these conifers will all throw their pollen up into the air, get blown around in the wind, and it'll land on the female cones of other conifers. There's no behaviour, and there's no morphology that can really control who's mating with whom. That means it's very hard to produce reproductively isolated groups, and therefore very hard to speciate. Now, animals, of course, get around this problem by employing morphological tricks, behavioural tricks, both in this case, quite dramatic ones, that result in reproductively isolated populations, and those then go on to form species. And the angiosperms have followed this trick to some extent. So by harnessing animals, which have behaviour, to carry their pollen around for them, to do this cross-fertilization, they have the opportunity, and maybe opportunity isn't the right way to think of it, because actually it might be a bad thing in many cases, but they certainly find themselves harnessed to the behavior of the animals that pollinate them. And so if those animals have different types of behavior, then that will result in different selective pressures on the plants and the opportunity to produce different flowers, which will then be reproductively isolated from other flowers who have been selected to fit the morphological or behavioral requirements of other animals. Now, there are lots of very good systems in which this can be studied, and this is just perhaps the best one, and there have been a couple of great talks on Petunia in this meeting. So in the Petunia genus, speciation has resulted from the recruitment of different animals as pollinators, so bees pollinating these purple ones, moths these white ones, hummingbirds these red ones, and the floral morphology has adapted to suit the behavioral requirements and the physiological requirements of the pollinators. So different colors are the obvious thing you can see, and that reflects the visual capabilities of those animals, but there are also differences in flower shape, scent, reward, and so on. So if you want to understand this species radiation and these relationships between plants and animals, I think you really want to think about it from 3 different angles, and that's what we try to do in my lab. So we take traits of flowers that make them attractive to different animal pollinators, and we try and explore their developmental biology. So I'm a developmental geneticist by background, and we try to figure out how these traits are built. We then try and explore why those traits are there, what it is they're doing functionally that makes them useful to particular animals, that makes them attractive to different animal groups. And I think of these 2 questions as the how question and the why question. So how is it built? Why is it there? And if you understand how and why, then what you can do is look in a phylogenetic context where, for instance, this trait is changing here and here, and ask yourself, okay, what's the molecular basis of that change? What molecular evolution has occurred? Because I know how it's built. And what's the functional or adaptive basis of that change? Have you got a shift in pollinator type, pollinator behavior? Why has that mutation been allowed and selected for? And so by looking together at the how and why questions, we can really get this understanding of the radiation across a phylogenetic tree like that. So we use this 3-pronged approach to think about various different traits of flowers that attract different sorts of animal pollinators. I'm going to focus today on 2 traits that you can see in this one single scanning EM image. So I'm going to talk later in the talk about the shape of the cells here. And this is an unusual cell shape for plant epidermal cells. They're cone-shaped or conical, and they're coming out of the screen towards you. Before I talk about that, though, I'm going to talk about these little ridges that you can see on top of the cells, and the way those little ridges interfere with the light in a way that attracts animals. So if you organize those ridges carefully enough, regularly enough, on a flat surface, they can produce color. And for those of you who were in a talk earlier this afternoon, you might have come across the idea of structural color, which is the idea of producing color by a transparent material that's organized in such a way that it interferes with light. These tend to be very ordered structures. So I'm showing you an example of that in this hibiscus trionum flower here. We saw these yesterday in Linnaeus's garden, and they were doing just the same as they're doing here, showing you their structural color. So you're looking at 5 different petals, each of which is primarily white with a dark red purple pigmented base. That's a simple anthocyanin pigment that selectively absorbs certain wavelengths of light. But the lower 2 petals, I hope you can see, look blue, and the upper 3 petals don't. So on that dark pigmented region, you can see blue down here. And that's an angle-dependent color. If I tilt the flower away from you, you would see the blue shifting up to the upper petals, and you'd see it lost from the lower petals. And an angle-dependent color is also called iridescence. So it's the same effect you see in a soap bubble or petrol on a puddle. And it can only be produced by a structural means. You can't produce iridescence or angle-dependent color with a chemical pigment. So these little ridges I showed you are producing this blue color by acting sort of like a diffraction grating, which is a fairly straightforward physics concept. And it's a diffraction grating that causes the iridescent effect you see on a DVD or a CD. So if you look at a CD, you can see the iridescent coloration. If you look at the CD on the scanning electron microscope, these are the data grooves here. And the data grooves are spaced at the same sort of frequency as a wavelength of light. And what that means is that the ridges between the data grooves act as a diffraction grating. And when white light arrives at that surface, it experiences interference, so constructive interference in some angles, destructive interference at other angles, and as a result, the light is split into different colors, and different colors are reflected at different angles. So you see red, yellow, green, blue at different angles. And what I've got up here is a picture of what happens if I make a transparent replica of that CD surface. So it's a perfect replica in a totally transparent material, and shine white light on it. And you can see that it produces color. And this is through this diffraction grating effect. Now, if you look at an iridescent flower, and this is actually a tulip rather than the hibiscus, on the same magnification as the CD, you see that those folds or ridges on the surface are spaced at the same sort of frequency, and they're working in the same way as a diffraction grating. If I make a transparent replica of the tulip surface, I get color, blue and yellow light. It's not quite as good. I'm not getting all this range of colors. And that's because actually, if you're looking cross-section down there at the base, the tulip grating isn't as regular as the CD. It's biological, it's not going to be. We've got narrow gaps and wide gaps, and that affects the color that's produced and makes it less perfect. Now that disorder effect is probably actually very significant in how the diffraction grating works, and you can hear more about that if you come to a talk tomorrow morning in the epidermal section. So we have this diffraction grating that's producing color. And what I want to do is talk a little bit then about the development of that, how a plant, or any biological organism, is making a structure on that nanoscale so regular that it can interfere with light in this predictable way. Then I'm going to talk about the function, what it's doing there for the bees, and we'll try and put it together at the end and talk about the evolution. So to explore the development about this, we have to develop a model system. Most conventional plant models are not iridescent. You don't come across that many iridescent flowers. It's not very common. So we had to develop a new system, and the one we chose was that hibiscus that I showed you at the start. It's actually a reasonably good genetic model. It's quite fast from seed to seed in about 8 to 10 weeks. It makes big flowers. It's easy to grow in the greenhouse. And it has a really nice advantage for a developmental biologist in that within a single petal, we have regions that have the diffraction grating. So in the purple patch at the base here, we have these ridges on the cells, but on the white parts of the petal, the cells are cone-shaped and smooth. So we can compare the two regions within a single petal, and that makes life quite a lot easier for the developmental biology. Now, I need to tell you what the folds or ridges are made out of first before I can get into their development. And they're produced out of something called cuticle. So the cuticle is the hydrophobic, waxy material that all plant epidermal surfaces have on top of them, and without which, as soon as the sun came out, the water would get sucked out of them and they just dehydrate and die. So the evolution of cuticle was a fundamental, important trait when plants invaded the land, and all plants produce it. But it's quite a difficult thing to put together, because you need this big hydrophobic polymer on the outside of the cells. And so we know a fair amount about cuticle production from work in Arabidopsis and also tomato, where having a decent cuticle affects how long your fruit lasts in the supermarket. And so if you look here at two plant epidermal cells side by side, here's the cell membrane. This yellow bit's the cellulose cell wall, remember the rigid box that sits around plant cells. And the cuticle is this region out here on the surface. And the cuticle is made through a fairly complicated pathway. So a set of transcription factors called the SHINE proteins, they're an AP2 subfamily, activate transcription of a set of genes which encode enzymes which make monomers, my yellow circles, of cutin. Now those cutin monomers are transported across the cell membrane by a subfamily of ABC transporters, and then out here on the cell wall, outside of the surface of the cell wall, they're assembled to make the cutin polymer. Now to do that they need an enzyme, CD1, cutin polymerase, and it's also produced inside the cell, transported across the cell membrane, and does its polymerization out on the surface. To make things a bit more complicated, there's a second set of monomers that can be involved, wax monomers, my pink blobs, and these are activated again by a similar set of transcription factors, the DWAX proteins, again activating the enzymes, making the monomers, they're transported and incorporated in the cutin out on the cell surface. So our developmental question was, how do you build this thing out on the cell surface, and rather than just having it there and flat, you actually put this pattern of ridges on the top of it. And to start with, we thought this was a really difficult developmental question. We weren't quite sure how we were going to tackle it. But we turned to modeling to try and get some basic ideas about how this thing could be built. And the modeling has been incredibly useful for us. Our modeling is fairly descriptive, but it allows us to work out what elements of developmental biology might be important to consider in our work. And it also helps us to work out whether we've found all the things that are important, as I'll show you in a minute. So I'm going to show you how the model works in my head, which is very simply. You have to imagine you have a cell that's a young cell. It's just finished dividing. It's been growing equally in all directions at once. It's an epidermal cell, and so it's producing cuticle. And you have to think of the cell as having a stiff surface. It's a cell wall. It's cellulose. It's this stiff material. Now it's secreting a viscous, amorphous, softer material, which is the cuticle. I like to think of this as sort of treacle oozing out on top of the cell. And that material is connected. There's an interface between the two of them, but they are fundamentally separate materials. So we've got a soft material sitting on a stiff substrate. Now, at some point, the cell changes its growth pattern, it goes into differentiation, and it perhaps starts to grow anisotropically. And the cuticle will take a while to catch up, because its tendency will still be to spread equally in all directions at once. But because they're connected, there's an interface between them that introduces forces, and it introduces compressive forces and stretching forces. And if you take a soft material on a stiff substrate, and you compress it and stretch it, then it buckles. And those buckles, that buckling in principle, could be enough to generate my diffraction grating if it's ordered at the right level. So we started off with this very simple model. It actually looks like this, and it says that you've only got two terms that you need to take into account here, the stretch and the cuticle synthesis. So the lambda terms here are stretch in the x-axis of the cell, stretch in the y-axis of the cell, and you can move within a graph between different colors, which mean different patterns of cuticle, simply by changing the amount of growth in one or other of those axes. So you can move from an irregularly patterned cuticle to a diffraction grating simply by increasing the stretch in one axis of the cell. We've also got this beta term here, so the two different graphs have two different beta terms, although obviously it's a continuous term, and that's amount of cuticle produced, the thickness of the cuticle. And so again, you can move from one cuticle pattern to another, not by changing cell shape, but actually by changing the amount of cuticle that's involved. So the model starts off very simple. And it's the same sort of model that underpins, for instance, how your fingerprints are produced by a similar buckling mechanism. So we wanted to test the model. We wanted to see whether it explained everything that we were seeing. And to test the model, we have to perturb genes involved in cell growth or cuticle synthesis in our hibiscus flower and see what the effect is on cuticle patterning. So that's what we've been doing, and I don't have a complete answer for you, but I'm going to show you a little bit of what we've done and a few snapshots of data. So here's my model of cuticle synthesis again, because I'm going to start there. And we could, in principle, perturb any of the components, but we decided to start at the beginning and alter the amount of production of the shine transcription factor, and that's going to alter the amount of cutin monomer produced, and therefore the total amount of cutin. And we developed an experimental design that is essentially quantitative. So we designed a battery of transgenic constructs here that should give us lots of more cuticle, a bit more cuticle, a bit less cuticle, or a lot less cuticle. It's just using different combinations of promoters and activation and repression domains. And we've introduced all of those into our plants, so we should have this quantitative range of cuticle production, which we can then relate to the patterning we see and feed back into the model. And I'm just going to show you one data point, which is what happens here in the plants that are ectopically expressing shine and are making a bit too much cuticle. And what we see on tissues that don't normally have anything other than a smooth cuticle, so the back of the petal and the leaf, we start to see ridges developing. And so that's sort of proof of principle, that if we change the amount of cuticle, we can change the patterning of cuticle that's produced. It fits with the model. But we need all the quantitative data before we can be sure the model is accurately describing what we see. It's clear it isn't describing everything, though. One of the great things about a model is that you can ask whether it really shows, really explains everything you see just in the wild-type situation before you even start doing experiments. This one doesn't, because our model predicts that if two cells have the same shape and the same amount of cuticle, they should have the same pattern of cuticle. If the only factors that are important are stretch and cuticle production, then that follows. But in fact, if we look at a cross-section of our hibiscus, the cells of the upper epidermis here and the lower epidermis have exactly the same shape, and they have exactly the same amount of cuticle, which we can measure by staining it here and measuring the depth of the cuticle. But the patterning of the cuticle is different. You can see the ridging on the upper epidermis, and there's no ridging on the smooth lower epidermis. So there's obviously a factor we're not taking into account. And what that factor is, the clue to it, actually comes from this staining. Because the two epidermal surfaces have taken up this stain very differently. The upper epidermis is much darker red than the lower epidermis. It's actually the same microscope slide. And they're taking up the stain differently because the cuticle has a different chemistry. And that made us realize, of course, that we were missing a key component in our model. We're assuming that we've got a soft material buckling on top of a stiff substrate, but we're assuming that that soft material is always the same degree of soft. And actually the softness or stiffness of the cuticle is going to affect its ability to buckle, and that softness or stiffness is going to be affected by the chemistry. So we potentially have a system here where actually, as well as cell growth and amount of cuticle, the chemistry of the cuticle is going to drive the buckling. Now, to explore that, we needed to find methods of analyzing the chemistry of the cuticle very precisely on different parts of the petal. And so, working with colleagues in the chemistry department in Cambridge, we used this technique called laser MS, liquid extraction surface analysis, to basically drop a drop of solvent onto very precise parts of the petal and then suck it straight back up again and apply it to a mass spec so that we could determine the components. And we were able to do that with the ridged region and the non-ridged region within the same petal. Remember, that was one of the advantages, that we have both cell types present, both cuticle pattern types present on a single surface. So I don't want to worry particularly about the detail, but just to show you that we compared these two regions, the ridged region and the smooth region, and that dark purple squares on this plot are compounds that are only present in the ridged region of the petal, and white circles are compounds that are only present in the smooth region of the petal. The graph is basically dividing things up by the amount of carbons in the chain and the amount of oxygenation they have, and you can see that there are some very specific compounds present in the ridged region, and indeed some very specific compounds present in the non-ridged region. The chemistry of the cuticle is clearly different. So that fits with the idea that the chemistry of the cuticle driving the stiffness, the Young's modulus, is likely to be affecting the way the cuticle buckles. And so to go back to that, we need to think then about perturbing the chemistry of the cuticle with our transgenic approach. We decided to focus to start with on this polymerase up here, CD1. By varying the amount of the polymerase in the same quantitative way I've shown you with the transcription factor family, we should be able to vary the stiffness of the cuticle and therefore see how that affects the buckling. And so again, just a single data point rather than the whole quantitative set. Here's what happens to the petal, wild type with all those long, thin ridges on top of the cells. If we overexpress the cutin polymerase, we lose the ridging completely. So if we make the cuticle stiffer, we appear to lose the ridges, which again fits with the idea that the chemistry here is driving part of the patterning that we see. So we've got lots more work to do here on this. I really want a quantitative analysis on both fronts and to be able to patch that back into the model and iterate it. But I hope you can see how we can use this sort of transgenic approach combined with the modeling to tackle the development of a trait in a originally non-model species. I'm going to change tack and tell you a bit about the function then now, about what these ridges are doing, why my hibiscus might bother making these ridges and this iridescent color effect. And this involves a very different set of experimental approaches. So I think this is probably the lowest tech experimental setup in Cambridge. It involves buying a box of bumblebees. They're sold for pollination of tomatoes and strawberries in greenhouses, connecting it with a gateable tube to a plywood box with a perspex lid that we call the flight arena, and letting the bees out to forage. So when they come to us, they've never been out of the box, they've never met a real flower. We can train them that anything we like is a flower, and so our standard training flower is a sterolin tube with the lid painted pink and an upside-down Eppendorf cap on top with sucrose solution in. And they come out of the colony, they forage in this flight arena for what they think is nectar from these cups. They take it back to the colony to feed to the larvae and the queen. While they're out foraging, we can paint mark them, so I hope you can see there's a blue spot here and a red spot here, and that lets us identify individual foraging bees. And so we can get some very robust data that's easy to analyze statistically. We can ask, for instance, what the first 100 choices made by 20 independent individuals is. And so it gives us good, statistically robust data. Now I'm going to show you just two experiments that we've done to try and explore how this diffraction grating might be affecting bee behavior. And the first is an experiment that asks whether a trait makes a flower more salient, more visible to an insect, makes it easier to find. And so the experimental setup is that we have 3 artificial flowers equally spaced in an equilateral triangle in the flight arena. They're spaced sufficiently far apart that the bee can't see them unless it's up in the air, so when it's landed on one, it can't see the other 2. And they all have sucrose solution in the cap in the middle. And what we simply do is time how long it takes the bee from leaving flower 1 to finding flower 2, from leaving flower 2 to finding flower 3. Of course, it doesn't matter which way around the triangle she goes. And we can do that repeatedly for an individual bee, and then for many different bees, all for iridescent flowers, and all for non-iridescent flowers, and compare the effect we get. So here's a data set looking at red iridescent flowers. The 2 points on the graph are how long it takes the bee to make that first flight from flower 1 to flower 2, and then the second flight from flower 2 to flower 3. So 4 to 5 seconds, essentially, to find a flower. Now, if we then add a diffraction grating to the surface of the flower, and in this initial experiment we've done that with a perfect diffraction grating that's copied off a CD surface, so we've got iridescent flowers here, you can see that it's significantly quicker. It knocks at least a second off the foraging time. And I think that's through the simple and obvious effect that if I dropped a brown button on the floor here and was looking for it, as I moved my head, I would catch the different colour flashes of light, and it would make the flowers stand out more, the buttons stand out more. The flowers are more salient to the bees for the same reason. But the real question is, that's not what a a real flower looks like. I showed you that the tulip is not as good as a CD at making iridescent colours. What happens if I put the surface of a flower, of an iridescent flower, onto these red discs rather than the surface of a CD? And the answer was really quite surprising. So here's the same experiment done with the tulip surface on the red disc. Again, exactly the same setup, and you can see that the floral diffraction grating is just as good, if not better, than the artificial one at catching the bee's eye. It makes the flower more salient to the bee, speeds up the foraging time. That means the bee prefers the flower because it's more efficient to get the food out, and that's, of course, an advantage for the flower. So floral iridescence significantly increases flower salience and bee foraging speed. But that might come at a cost, because a foraging bee doesn't make individual decisions every time it approaches a flower. When it finds a rewarding flower on a foraging bout, it forms an image of that flower, we call it a search image, and then it continues to forage for other flowers with that same search image throughout that foraging bout. That's good for the bee. It doesn't have to make too many difficult decisions in any one foraging bout, and it's good for the flower or the plants because it tends to mean pollen moves between individuals of the same species. But you can imagine that being iridescent might compromise that. If you look different colours from different angles, it's kind of hard to form a fixed impression of you that's consistent. And so this is a second experiment to test whether iridescence actually compromises search image formation and makes it hard for bees to find the same flower. So what happens here is the bee is allowed to forage for at least 15 to 20 foraging bouts, that's usually about 150, 200 flowers, on artificial blue flowers that all have sucrose in them. So she should form a really strong image that blue is good. And then we introduce a couple of distractors. So these are fake flowers that are a similar colour. They're far enough apart in bee colour space that the bee can see them as different, but they're not easy to see as different. It's not blue versus yellow. It's quite a close colour. And we ask whether she can identify that these flowers are different. And we do that same experiment with non-iridescent flowers, and we do the whole experiment again where all of the flowers are iridescent, and ask whether the iridescence affects how easy it is to form the search image. So here's the data set, and you're looking at correct choices out of 20. So when the flowers are not iridescent, the bee gets it right about 70% of the time, 14 out of 20. So it's quite hard to do. If the flowers were completely different in bee colour space, I'd expect her to get it right 90% of the time. But she's certainly doing it more than 50-50. If we add an artificial diffraction grating from the back of a CD, that drops to 50-50. She really can't correctly identify blue flowers against purple flowers if they've all got a perfect diffraction grating on and they're all iridescent. But again, the interesting thing is what happens when we repeat the experiment with a diffraction grating from a flower. Same result for no iridescence, but the floral diffraction grating has no effect on the bee's ability to form an accurate search image. It doesn't compromise search image formation. So the floral diffraction grating was just as good as a perfect one at making the flower visible, but just as good as no diffraction grating at making the flower easy to remember and identify for the bee. And so perhaps part of that disorder we're seeing is actually not just accidental, it's optimized to speak to the target that flowers are signaling to. Now, floral iridescence is so good, it's perhaps not surprising that it's reasonably widespread across the angiosperm phylogenetic tree. It's not a common trait. You don't see that many iridescent flowers, but we've looked through all of the major clades of the angiosperms, and we find it in almost all of the major groups, except the early diverging ones, and present in many species within these groups. So although it's not very common, it's phylogenetically widespread and looks like it's evolved convergently many times. And of course, that's the next thing that we're looking at, is whether that convergent evolution is through the same developmental mechanisms as we're seeing in our hibiscus. Okay, so I've told you a little bit about the tiny ridges on top of the cells on top of the petals and how they affect pollinators, looking at it from these three angles. And I'm going to change tack again and talk now about the cells themselves and the shape of those cells and how they affect pollinators. So if you remember, I showed you earlier that the cells on a petal epidermis are this unusual cone shape. If you look at a leaf on the scanning EM, the cells are generally flat. And yet this morphology is probably the most common morphology for any flower petal. Something like 3/4 of petaloid angiosperms have conical cells on those petals. So I'm going to skip very quickly in this part of the talk over the development and the function, because this is much older work, and really focus on the evolution. So I'm going to try and tell you how they develop in one slide and what their function is in one slide. Hope it works. This is the developmental slide. So this is work that was done a long time ago. We essentially know that conical cell outgrowth is regulated by a MIB transcription factor. The R2-R3 MIB family is much expanded in plants compared to MIBs in animals. And the MIB transcription factor that regulates that cell growth does so by controlling outgrowth in the Z plane, so that's towards you. and it does it very specifically in the petal epidermis. The gene's called mixture, and it was isolated from this antirrhinum mutant that has a transposon insertion in the locus. You can see on the unstable version here where the transposon is excising somatically, that in the wild-type cells where it's excised, you get the conical shape, and in the mutant cells where it's present, you lose it entirely, you get the flat shape. So it's the master regulator of the pathway. It's necessary for conical cell production, and we know from transgenic work actually in many species now that it's also sufficient to induce that conical cell outgrowth. So that's the development. It's conveniently down to a single transcription factor. Here's the function. The function was quite hard to work out because actually those conical cells do a lot of jobs. They focus light into the flower, make it look brighter in color. They affect the temperature, they affect the wettability. But the thing that matters to the pollinators is that they affect the grip. They affect how easy it is to get a grip on the petal when you land. And so here's one of many experiments I could show you that shows that point. What we've done here is take those wild-type antirrhinums and the mixture mutant that has flat petal cells and offer them to the bees in two different orientations. If we offer them horizontally, so the bee has to land on this horizontal surface and stick its tongue in this tube to get the nectar reward, and we ask, what's your preference for the conical celled line? The answer is 50-50. The bees don't care. But if we offer them in traditional antirrhinum presentation mode, so you've got to land on a vertical surface and open the lip to get in to get the nectar reward, then we see a big increase in preference for the conical cell form. And we can essentially repeat that set of data by making the flowers difficult to handle in a variety of ways, by changing the angle, by making them move as if in the wind, by making them wet as if in the rain. So when flowers are difficult to handle, bees prefer them to have conical petal cells, and you can see, looking at a bee on a normal, on an antirrhinum flower out in the field, that actually the claws here have to get quite a firm grip onto this vertical surface. Flowers aren't nice and easy and flat to land on, and the claws interlock into those conical cells. So it's a trait that's really useful. It's a trait that we understand how it develops. We have the opportunity to explore how it evolves. And the first question we wanted to ask was, how old is it? Is it a trait that's ancestral or ancient in the angiosperms, or is it something that's evolved repeatedly across the groups as the flowering plants diversified? Now, to do that, we went back to the base of the angiosperm phylogenetic tree to have a look at the plants down there. So most angiosperms are here, in the bit we're calling other angiosperms, but there are three orders down here right at the base of the flowering plants that are still extant. Amberolales is a monotypic order, and the single species that there is in that group has really strange derived flowers, so it's actually quite a difficult model to work with. But the nymphales here are the water lilies, and there are quite a few extant species of water lilies, and so we can look at what's going on there. And actually, Charlie introduced us to Kabomba here, which is a nice model water lily, small and easy to grow in a fish tank. They're often sold for goldfish tanks. And it has very beautiful conical cells, as you can see here, on these petal-like structures. Now, they're not actually petals, they're undifferentiated tepals, but the principle is the same. They're the insect-attracting organs, and they have these lovely conical cells. So we wanted to know whether those conical cells, which appear early on in angiosperm evolution, are produced by the same pathway. Are they regulated by mixture? And to do that, we needed to find the mixture gene in Kabomba. Unfortunately, there is only one. This is a gene family, a subgroup of the MIB transcription factors that can have half a dozen members in some angiosperms, but there only appears to be one in Kabomba. And we had a look, just a correlative look through quantitative RT-PCR at its expression profile relative to growth of those conical cells. So you're looking here at buds getting older to my left. This is the youngest stage at which the conical cells just start to form, and we're looking at expression in tepals, and then the stamens and carpels, the reproductive organs. And you can see that there's massive expression of that gene in the developing tepals at the stage when conical cells are starting to appear. So that's good correlative evidence. We can't do transformation in Kabomba, but what we can do is test whether that gene, the Kabomba mixture gene, is capable in another tissue, in a flat cell tissue of a transformable plant, of inducing cell outgrowth. And so we do that by expressing it, in this case in tobacco, where the ovary cells are flat, the style cells are flat, but if we express Kabomba mixture in them, it causes cell outgrowth. And so it's correlative, but it's quite good correlative evidence that this is a gene that's expressed at the right place at the right time and can induce epidermal cell outgrowth and is likely causing the production of those conical cells on the tepals. So if that's the case, looks like conical cells are pretty ancient in the angiosperms. They're pretty useful. But actually, I said at the beginning that about 75% of flowering plants have them. The rest don't. And so why wouldn't all flowers have conical petal cells if they're so useful? And that question has been the focus of quite a lot of work in my lab over the last few years. So again, I'm just going to show you a couple of snapshots of answer to that. One part of the answer is that if you switch pollinator type, then maybe conical cells aren't quite so useful. So for instance, in Nicotiana, the tobacco genus, there are at least 2 independent losses of the conical cell form associated with a shift to pollination by hawk moths. And of course, they don't land. They hover when they're feeding, and so grip probably isn't as important to them. It's a similar story around Antirrhinum itself. So this is Rhodochiton, and Rhodochiton is a close relative of Antirrhinum that shifted to hummingbird pollination and has lost its conical cells. And again, hummingbirds are hovering pollinators, not landing pollinators. So as you change pollinator type, as we discussed right at the beginning, the selective pressures on the flower change, and you can see that you might have changes in morphology as a result. But I want to finish by just telling you a very short story about how you can also get this change in morphology, not as a result of shifting pollinator type, but just as a result of shifting pollinator behavior. Because one of the great misconceptions about plant-pollinator interactions is that everything comes down to one plant, one pollinator, that it's all about species-specific specialist interactions. And actually that's not the case at all. Bees will visit any flower that looks like it's got a decent amount of nectar in it, and many flowers are pollinated by many types of insects. And so the selective pressures can come not just from who pollinates you, but how they interact with you. So this is a story from the genus Solanum, which is the potato, tomato, nightshade genus. And Solanum has a very unusual pollination system. It doesn't produce nectar as a reward for its pollinators. It produces far too much pollen. So the pollen is produced in these really big, elongated stamens or anthers here. And instead of dehiscing like a normal plant, where they peel open and the pollen's exposed, these anthers dehisce through pores, which you can just make out here in the tip. What happens is that the bee comes along, foraging for pollen, which is very high in protein and is used to feed the larvae, grabs the anthers and vibrates its flight muscles, and the pollen flies out and lands on the bee's body, and then it can be groomed off and fed to the larvae. So this is a method of pollination called buzz pollination, and the entire genus Solanum is buzz pollinated. Now, it's a big genus, but if we look at this cartoon phylogeny of it, each of the arrows here is showing you an independent loss of the conical cell form across that genus. So in this big genus, we're repeatedly losing conical petal epidermal cells. And if I focus in a bit more, I can show you how we're doing that. So it's really just a case of taking bits of the phylogeny, scoring the petal cells as flat, which are the black boxes, or conical, the white boxes, no data yet if there's no box, and then doing some analysis to see when the conical cells are lost. So you can see, for instance, that Solanum capsicoides down here has clearly lost its conical cells independently from this big group up here, which have probably lost it somewhere along the backbone. Then what we can do is take these independent losses of the conical cell form within the genus and ask what's the molecular basis of that evolutionary loss. We know how you make conical cells. Are they being lost repeatedly, convergently in this genus through the same mechanism or through different mechanisms? So here's some data from just 2 sister pair comparisons. That's Solanum capsicoides that I just showed you. It has flat petal cells, and here's a close relative, Solanum cecimbrifolium, conical cells. And then in a completely separate bit of the phylogeny, the kangaroo apples, these are Australian, here's Solanum aviculari with flat cells, and here's Solanum laciniatum, which has conical cells. And so what we've done is look in those 2 sister pairs at the expression and activity of the mixed-alike genes. Now, I said this is a small group of transcription factors. There are 6 in Solanum. But we've been able to identify one of them that's consistently expressed in petals and involved in this conical cell outgrowth. And this gene, if you look again, qPCR, very simply, the dark bars are the flat-celled relative and the light bars are the conical-celled relative. So there's the kangaroo apples, and the conical-celled one has much more expression of the gene. Here's Solanum capsicoides, and the conical-celled cecimbrifolium has much more expression of the gene. So we're seeing repeated loss of the conical cell form through repeated loss of expression of the master regulator of the pathway. Of course, the next question is what's causing that loss of expression? Is it changes to the regulatory regions of the gene itself, or is it something happening upstream? And we're working on that at the moment. But it's nice to be able to see how this convergent molecular evolution within a genus is happening. The other question is, why is it happening? Why would you repeatedly lose conical cells within this genus? So we designed a fairly complicated experiment with our bees to explore this. And we made these very high-tech artificial flowers. They've got 5 petals, and the petals have the surface impression of a real petal on them. So they either have conical cells or flat cells. And they have a hole in the center here, out of which is protruding the real reproductive structures of a real Solanum flower. So you can see the 5 big yellow anthers here that produce pollen, and the carpel actually is just sticking up there. And so we had these flowers with conical cells, we have them with flat cells, and we asked the bees, if you're foraging for pollen in this setup, what's your preference? And we set out to find out whether they preferred conical cells or flat cells. In fact, it was one of those experiments that it took a very long time to design and almost wasn't worth doing, because as soon as we started it, we could see what the answer was going to be. The bees have absolutely no preference whatsoever for conical cells or flat cells in this system, and I can show you why. If you no, I can't. I ought to be able to show you why. If you watch the bee interacting with the flower, apart from the odd slipped foot, she doesn't touch the petals at all. So this is a pollination system which is entirely focused on the stamens. She's only gripping onto those. She's actually using her claws to grip into the stamens themselves. And apart from the odd slip, she doesn't contact the petals. So it's not surprising that she has no preference for conical or flat-celled flowers. So within this system, we're seeing repeated convergent loss of conical cells through parallel mechanisms, at least parallel loss of expression of the transcription factor that regulates the pathway, probably as a result simply of genetic drift, that there's no selective pressure to maintain the conical cell type, although possibly there may be some cost associated with making conical cells, either because of the metabolic cost of making them, or actually because they do provide grip, they might distract the bees' feet a little bit from the anthers, where actually they're most needed. So that's something we're still working on exploring. Okay, so I hope I've shown you a little bit about how we take this sort of three-pronged approach, thinking about development and function, to get to a sort of integrated understanding of evolution. We do it with lots of other different floral traits, which it would be great to talk to you about if you're interested. But I've shown you it with a couple of different aspects of the petal epidermis. And I need to finish, as usual, by thanking the people who actually do the work, which really isn't me. I've talked today particularly about the iridescence work, which is done by Edwige Myrood here, who's a postdoc in the lab and who's sitting down there. Thank you very much. A lot of our work is done in collaboration with Sam Brockington at the back here, who does a lot of our phylogenetic reconstruction with us. The work is collaborative, and I work particularly with Paula Riddell and Richard Bateman at Kew on these projects. Marcus Calborough at Cambridge is our chemist who helps with the chemical analysis. Silvia Vignolini helps with optical analysis. She's a physicist interested in structural colours. Thank you very much for listening. Arguing about who's in charge. Maybe I can be in charge. Does anybody have any questions? No, they all want to go to the bar. Oh, sorry, Charlie, I didn't see you.
So the conical cells arise through the mixture mechanism. They're present in really basal angiosperms, and that's a MIB, so a big family. What was the pro-ortholog of mixture doing prior to the angiosperms?
Okay. It's a good question. If you were at the protein evolution session this morning, I'm not sure I dare talk about the ortholog of mixture and anything before the angiosperms. The subgroup of the MIB family is clearly there. So there are members of that subgroup all the way back through land plants. We have not looked at the rest of them, but we have gone back to Marcantia in the liverworts and looked at what the gene's doing there. So if I take the Marcantia, the liverwort gene, and of course that's separated from the angiosperms by nearly 300 million years, more than 300 million years, and I express that in tobacco, it causes cell outgrowth just the same way the Kabomba mixture does or an Antirrhinum mixture does. So the protein function is really highly conserved. What it's actually doing in the liverwort, I'm not so sure. So we have overexpressed it in Marcantia, and it increases the number of papillae you get around the antheridia, but nobody really knows what those papillae are doing. I'd like to see a knockout phenotype, and we haven't managed that yet, so I'm kind of not putting any money on it till I see that.
Yeah.
Hi.
Yes. Thanks, Beverley, for this great talk. So just a quick question about the regularity on the structures, or actually the lack of regularity in some cases. So, as you know, some biological systems can generate extremely organized and regular structures.
Yeah.
And so this question of noise is super interesting, and it's very difficult to know how to tackle that question. Is it useful indeed to be disorganized a little bit? And I would like to have your opinion about this in the case of the flowers.
So you're asking me to preempt a talk tomorrow morning by my postdoc. I think it is useful. I think it is useful. We started off assuming that it was just necessary, that it couldn't generate something on the nanoscale so perfectly ordered that it didn't have this degree of disorder to it. But actually what we see looking across that convergent repeated evolution of the gratings is that they always have the same sort of amount of disorder in them. It varies in whether it's in the period or the height, but it's always the same sort of amount of disorder, and it always sort of converges on the same sort of optical signature. So I suspect that actually there is some purpose to it, but I will let Edwige tell you more about that tomorrow morning.
Okay, I think we should draw this session to a close now. Thank you very much indeed, Beverley, and thank you all for coming. Ed. Thank you.